And for that, it's really wonderful that Maurizio Corbetta is here. Maurizio, of course, is well known for his work in neurology, in Connectomics, and also his application to the different deficits due to stroke, where he has advanced a very particular, that's a whole brain perspective, using a range of techniques, including fMRI or EEG. So he moved back from the States, and now he's running the neurology department at the University of Padua since a few years, where he's embarking on a huge project to sort of revolutionize how we do neurology and its applications in many forms, in particular in Padua. And we're looking forward to what you have to tell us. So welcome to BCBT.
Thank you very much. Okay, so this is a school, so feel free to interrupt me if there is, questions or if I'm not clear. So what I'll try to give you is a story, a perspective. I think that I hope is a bit different from, although it's kind of strange now. Most of you now are growing up with this idea of connectomics, and it seems like it's the last thing that has been appearing on the planet Earth. Well, this is actually the most recent evolution of a very long story that goes back almost 200 years. So I'm a little older, so I guess I get a little bit of this historical memory. So, and the story I'm going to tell you is a story starting with a straw man. Okay, most of you will not agree with it, but I think this is still the dominant theory in neurophysiology, that the brain is a device to analyze stimuli and plan responses. Okay, the idea is that basically you have what provides energy in the brain are the sensory stimulation, the sensory stimuli that are entering the brain. We have a set of detectors, specialized detectors, for instance, in the visual cortex. This is work by Hubel and Wiesel in the 1950s, showing that you have specialized cells that are analyzing specific aspects of the stimulus. And so we're building through this detector complex and more complex representations. You go to higher order visual cortex, where you build up representation of complex objects like faces or places. And of course, people have found specialized regions for these attributes. And then from there, this information gets transformed through what was called sensory motor transformations of different sorts, retinotopic, bodytopic, egocentric, allocentric, all of that. And then you get into the motor system, the premotor systems, and now you're planning responses for different effectors. And of course, you have specialized system for these different responses. So this is kind of the way we've been thinking about neuroscience in the last 30 years, 50 years or so. And from there, of course, it is logical that if you have lesion in different parts of the brain, then you will get different specialized deficits, because you have neurons that are specialized for different attributes. So these are three famous cases that are kind of cornerstone of clinical neurology. The first one is Phineas Gage. Is a guy that is a guy that received that rod, that steel rod, in his. It was a who knows the story of Phineas Gage? Okay, everybody. So basically, that rod ended up in his frontal lobe, and he woke up. He was fine. He was not, he was not unconscious. He could speak. He could move. But of course, his behavior was completely derailed by the lesion. From a church-loving, going family man, he became kind of a brawling alcoholic, spending all his money gambling. And so that idea that the executive function, the decision-making, now was localized in the frontal lobe. That was one of the the second guy is Victor Leborn, which was the original patients of Broca. So that is the brain is still at Paris 5 at the Museum La Salle, there is a museum where you have this brain and you can see it, and there was basically a lesion, as you can see, in the left frontal cortex, and this patient had a specific impairment in speech production. Okay, could just say a particular syllable, ton, but it could understand everything. And so there was the idea that the left frontal lobe was specialized for speech production. And the last one is, of course, the patient HM, who had a bilateral hippocampectomy and amygdalectomy, and basically developed this very profound amnesia. And HM, of course, is one of the kind of major cases in neurology where you have that the hippocampus is specialized for learning, for memory. Now, more recently, and of course this is work by Michel Thibault-Deschott, and we have seen that these lesions effectively are not just focal lesions of one part of the brain, but in fact there is a very complex pattern of disconnection, structural disconnection in this case, that is affecting each one of these three cases. So that from a very strictly modular perspective, now you're starting getting into this more system perspective. But still the case that we think that the brain is highly dimensional, of course, and that the behavior is highly dimensional, okay? At least in Western philosophy, we have this idea we're all different and we're all special, okay? In fact, in the Eastern philosophy, it's quite the opposite. We're all the same, okay? Where everybody is more similar to other people. And my perspective over the years is really changing. I think psychology, in a way, has done a disservice by emphasizing tiny difference between processes, while we've been completely ignored the similarity among processes, okay? And one of the major messages out of this talk, that there is a lot of correlation, not just in brain activity, but there is also a lot of correlation in behavior, normal behavior, as well as abnormal behavior across subjects. And so, although there is this wonderful complexity at the microscale, with billions of neurons and trillions of connections, which we've been emphasizing over the years, it is the case that when you look at the mesoscale, in fact, the story is much more simpler. And in fact, there are very specific patterns of correlation at the mesoscale level, which appears to be low dimensional. Okay, so that's an example of resting state fMRI, where you have regions of the brain, in that case, that are correlated with the primary motor cortex. And when you stare at these movies for hours, like I've done, you see emerging patterns of spatial temporal patterns that are emerging. And it's not like there are like hundreds of them, it's more like there are tens of them, or maybe a few. And this is now ECOG data. This is a patient that has been implanted with an ECOG grid, and now you're recording activity in the same kind of range as the bold, 0.7 hertz. And the little dot there is in the primary motor cortex. And you see that over time, the primary motor cortex show correlation, okay, the central sulcus and the precentral gyrus. And of course, you see a lot of dynamic correlation with other regions in the prefrontal cortex. And this is kind of the idea that is emerging, that you have this spatio-temporal pattern of activity that at the mesoscale level appears to be relatively low dimensional. And of course, the low dimensionality is related to the highly anisotropic organization of the structural connectivity, where you have now, of course, the white matter is not one big blob with fibers going everywhere, but there are very specific rules, spatial rules in how the white matter is organized, which is probably responsible to some extent, maybe 30-40% responsible for this kind of similarities that we see in correlation. And of course, people have known for a long time, and this is an old paper that I love by Engel and Singer in 2001, this correlation are present everywhere, okay? I was yesterday at here in Barcelona talking with Mavi Sanchez, and she showed me some slice data showing these intrinsic correlations in a piece of tissue that is completely disconnected from the rest of the brain. So you have basically activity that is intrinsic to the way a cortical column is organized. And now if you replicate that organization over many, many thousands and millions of columns, now you are this with some modulatory system, of course, then you have this very complex organization. So this correlation has been found at the level of single slices, it's been found at the level of local field potential, it's been found at the level of the whole brain. And so you see here is emerging a story. And one of the stories that has been, I think, most influential is that out of this pattern of correlation emerges a structure that is also low dimensional of how the cerebral cortex is organized. This is work from David Van Essen and the Connectomics project, in which out of 1,200 subjects that have been scanned repeatedly with functional MRI, with a completely data-driven analysis, you have basically 5 systems correlated and nested in the brain. Now, there is no real reason why you should have, 5 or 7 or 12, but the point is, it turns out that from a data-driven analysis, you have a very simple principal organization. You have sensory regions, visual regions, sensory motor regions, and auditory regions. And then you have these lighter gray and darker gray set of regions that have to do somehow with internal processes. And we're talking things like the default mode network and the frontoparietal network, and external processes, things like the dorsal attention network, the ventral attention network, and so forth. So you see, again, in healthy subjects, you have this low-dimensional organization. Now, when we go back to neurology and lesion, then, the thinking about the focal lesion has really changed. So from the idea from neurology, classic neurology, Phineas Gage and HM, in which you have basically the brain is organized as a set of experts. You have modules that each one is taking care of a particular function. In cognitive psychology from the 1960s to the 1990s was this idea that the brain is a set of, an ensemble of cognitive operation or mental operations, okay? This idea that you have, the analogy that I do usually is that a bunch of paper pusher. You have an office, like a corporation. Nobody really knows what's going on at the global level. Everybody's doing his little work in his cubicle. And basically you got an input coming in, a piece of paper, you do what you have to do and you move it out to someone else. And that's the analogy in cognitive neuroscience, that you have this mental operation, they're stringed together to generate complex behavior, okay? And of course, out of this, in terms of brain organization, then you have different ideas. So one is this modular view where every region is specialized for a particular function. And I can tell you that this local function view, although seems like so old, in fact is what we're still teaching neurology residents these days. So if you go in a neurology specialty program, they will teach you that, there is an area for reading, there is an area for writing, there is an area for speech, and we teach residents how to localize this disease, these disorders, based on the place in the brain where is the lesion, okay? Then in the last 25 years, we had cognitive neuroscience with this idea of networks, okay? You have the attention network, the motor network, the language network. So this idea of single operation, mental operation strung together to create complex behavior. But in this new world of connectivity, then of course, we're losing very rapidly the idea that there are modules where the neuronal properties are specific for a specific function, okay? I don't believe personally that we have neurons that are specialized for faces or numbers or other things. I think what is going on is that, of course, is the set of input and outputs into a particular region that is determining its level of organization. And so this picture here down here, you see that we don't have a motor system, we don't have a visual system, but we basically have a set of regions that, because of their properties and their connectivity with the periphery or with the central connectivity, now assumes a set of functional properties, okay? And this is, I think, best exemplified, for instance, by experiments in, deprivation experiments, where you take a patient and you blindfold him for a week, okay? And now you see that the visual cortex is dynamically changing its properties, and now it's beginning to respond to auditory and tactile stimuli. And so if you take this perspective, which is kind of the modern perspective on lesions and behavior, I think it is clear that when you have a lesion in the brain, you will not have individual deficits, and so you don't have double dissociations like the one we've been looking for in cognitive neuropsychology or neuropsychology for 30 years. But what you have a set of correlated deficits, okay? Because a lesion is going to impact a node, not belong to a particular network, but actually belong to multiple networks, and therefore patterns of correlated deficits should arise at the population level as you're looking at many, many patients. So a lesion like that may cause, for instance, deficits in maybe sustained attention, language, as well as maybe motor deficits, okay? So is there any question about this introduction? Does that make sense? That we go from...
You make a lot of assumptions about coding in this case, right? Because you're saying, look, it's not one-on-one, neither for neurons or networks, but it's like mixed.
It's not one-on-one, and it's not one-to-many, and it's also not many-to-one. It's a combination thereof. So a region can be part of a particular function, but can also be part of many functions. And it all depends on how that region is being synchronized or correlated with other regions. And I'm not saying that there is an innumerable number of possibilities. The possibilities are low dimensional, so there is constraint of how a region is actually getting synchronized. And but it's clearly not involved in only one function. And that's, for me now, is well clear, and I can show you.
Give us the data.
Yeah. Of course. Yeah. Okay, so in the rest of this talk, after this very long and winded introduction, okay, I will tell you a few things. I'll tell you about some data. We have been working on this project for now almost 8 years, okay, so we have quite a bit of data. And I'm going to go back to kind of the drawing board and ask, okay, if I got lesions in the brain, all right, let's suppose that I'm not a neurologist, and let's suppose that I don't know anything about neurology. Okay, I go back, and if I have a lesion in the brain and I get lesions one after another, how many neurological syndromes or pattern of neurological impairments am I going to get? Okay, if neurology is right, I should get a very high dimensional pattern of deficits, many syndromes. I can get Broca's, Wernicke's, optic ataxia, agnosias, apraxia, idiomotor, all of that. If if this new perspective is true, the pattern of lesions and syndrome should be quite small. Then I'm going to ask, what are the patterns of structural and functional disconnection produced by these lesions? And then the third question, which is still unanswered, how can we restore these abnormal patterns? Because of course, rehabilitation is a black box. We put in inputs and we get out outputs, but typically we're not looking at the representation in between. And I think this is where we should be going with neurorehabilitation next. All right, so these experiments are very simple. We take any newcomer to a stroke unit. This is a tertiary stroke unit in the States, and now we're continuing this work in Padua. So the patients are representative of the stroke population. We rule out all the people that have multiple lesions. We take only single lesion and first-time strokes. And then we rule out all the people that have what is called white matter disease, which is basically tiny little lesion in the white matter that basically making they're affecting the communication. So you get a patient with a lesion here. Now we provide them with a very deep, what we call both broad and deep neuropsychological battery and motor battery. And then we measure their DTI, and then we're measuring their functional connectivity. This is kind of the basic idea. We do it at the acute stage, and then we repeat this experiment again at 3 months and again at 12 months. And today I'm just going to tell you pretty much only acute data. All right, so the first question is, what are the most common neurological deficits post-stroke? All right, so here's the study design. In the first study that we published in 2015, we screened 6,000 patients to basically potentially enroll about 1,200, okay, which met criteria. And of those, only 10% participated in the study for different reasons. Some of them didn't want to participate, some of them were far away. We were centralizing patients with stroke from different cities in St. Louis, and so some people didn't want to stay with us for so long. And then, many reasons. So there is some epidemiological controls, which I'm not going to show you, that these 132 patients are actually representative of those 1,200 patients and of a larger database of stroke. Okay, so you have to trust me, because I don't have the time to present all the controls, but you can read the paper, that these patients have the same amount of diabetes, the same amount of hypertension, the same amount of cardiac disorders as a regular stroke population. And this is what we do at the acute stage. At 10 days, we get this battery of motor, vision, sensory, language, memory, and attention. Okay, we take anybody with any neurological deficit, and then we do a structural, functional battery and perfusion battery. And as you can see here, it's in those days was only 64 directional DTI. Okay, but we got 35 minutes of resting state. Okay, this is one of the things that I like to say for your school. Don't trust anybody that tells you that you can get 5 minutes of resting state and get a good connectivity matrix. Okay, you need at least 15 minutes. The networks, they stabilize within 15 minutes. Okay, 5 minutes, they're still very unstable. So in these patients, we got 35 minutes of resting state. And then we provide this data again at 3 months and in 1 year. All right, so these are some of the tests that we did. Of course, when you select a battery like this that you have to give to people that are acute, there are some trade-offs. So we decided to go with, say, regular kind of vanilla variety domains. Okay, we don't have social cognition, we don't have theory of mind, we don't have prospective memory. Okay, we have basic stuff. We have motor, language, attention of different kinds, egocentric, allocentric, sustained attention. And we also have visual memory and verbal memory. And the battery was selected for those of you that are neuropsychologists to kind of get into some of the dissociation that people talk about, okay? So for instance, we have tests for familiarity and tests for recognition in memory. We have egocentric and allocentric neglect in visual spatial attention and so forth. All right, so out of that battery and 132 subjects, we found that 3 clusters of deficits, 3 components, account for 70% of the variability. In other words, if I didn't know anything about neurology and I had 3 tests, I can describe 70% of these patients. And the clusters, the components that I see are this, okay? And the size of those balls is proportionally to the variance accounted for. We basically have one ball that is a cognitive language components, okay? These are deficits of language, verbal memory, and spatial memory. And the size of those letters is indicating the weights on the principal component analysis of those tests, okay? So you see it loads a lot on language and verbal memory and loads a little less on spatial memory. And then we have a left hemisphere component that is basically get right body deficits as well as some attention shifting and visual field bias for the right visual field. And now we have a right hemisphere component that gets a little bit of spatial memory, left body biases, visual field biases, which is egocentric neglect, and average performance, which is a form of sustained attention, okay? So as you can see, things kind of make sense, okay? You have a right hemisphere lesions, a left hemisphere lesion, and then something that is in between the 2 hemispheres, okay? Now, I can tell you that if you take the NIH stroke scale, which is basically a clinical battery, you also find 2 clusters, a left hemisphere cluster and a right hemisphere cluster. So at the population level, it is not true that the lesions are causing individual syndromes, but what we see pattern of correlated deficits, okay?
This is acute, right?
This is acute, 2 weeks. Now, those components that account for about 70% of variability at the acute stage continue to account for about 70% of variability. Of course, we decrease variance because people are getting better, and so the variability among patients is decreasing at 3 months and 12 months. So this is now a graph of the behavioral deficits where the line are basically expressing the correlation between deficits. And you see this very interesting triangular structure where spatial memory, visual memory is kind of a hub of this attention motor cluster and this language verbal memory cluster. And you see basically that the same structure is maintained over time. So as people are recovering, these clusters remain. And I think one interesting implication of this work, which has not been tested, is that if I want to retrain language or verbal memory, as well as attention, then I should probably start retraining spatial memory, because spatial memory may be the hub of interaction between these cognitive domains. Okay.
Wait, you say the clusters remain. Actually, if you look at the blue cluster, the graph
Yeah.
matches disappear.
Yeah.
Cluster seems to disappear.
Well, it's no. Well, okay. So this is the one that these guys are now coded as ipsi contra. Oh, I'm sorry. Yeah. No, that's okay. I use my. You see the arrow? Okay. Well, then we need a pointer. Okay. Yeah. So in this analysis, I keep separate I keep left lesion and right lesion separate, and so I get a left hemisphere cluster and I get a right hemisphere. Okay. If you keep the lesion separate, then the principal component works that way. I can also do ipsi contra. Okay, because motor deficit, you can code them as ipsi and contra. Now we're getting into the weeds here. So now I have one motor attention cluster, okay, that links to these memories, visual memory deficits, which in turn is common between this component and this component. Now at 3 months, as you can see, some of these links are below thresholds. Now here it is below, I think here the threshold is set at 0.3, this might be 0.21. But the point is, if I do a principal component separately for the 3 months data and the 12 months data, I always get these three components. Okay, now remember there is a difference between components and clusters. Okay, a PCA basically splits the variance and orthogonalizes the variance. It doesn't mean that there are clusters. Okay, now I use the word clusters in a kind of colloquial way. Okay, in terms of components. Okay, so in fact these are components in which I can divide the behavioral variance. Okay, now I can tell you basically this. Now it is true that as we go to 12 months, then for instance some of these links are getting below threshold, and so you can say, well, okay, the deficits are becoming more specific. So I get only, for instance, specific attention and motor deficits together as a syndrome, if you wish. Okay, but we have to move on. Okay, so in the last 2 years, as I got to Padua and I have a stroke unit and I have, now I can tell people what to do. It's easier to now collect the same amount of data rather than in 5 years, in 2 years. Okay, so in 2 years we collected exactly the same amount of data than in 5 years in St. Louis, and now we use a complete different battery. We use something called the Oxford Cognitive Screen, which rather than 2.5 hours, takes about 15 minutes. And so the Oxford Cognitive Screen is a battery developed by Glenn Humphreys. Unfortunately, Glenn died 2 years ago. And now we use the NIH stroke scale, which is the clinical scale that all the stroke doctors use. And so these two together, we did exactly kind of the same experiment in 2 time points, one within 1 week and one within 3 to 6 months. And again, by doing a principal component, we find, again, the same 3 components. We see a first component which loads on kind of left verbal and memory deficits. A second component that loads in deficits of the left body as well as attentional deficits. And then a third component that loads on right motor deficits. And when I compare the amount of variance accounted for in the Padua Stroke Project and in the St. Louis Stroke Project, you see basically that the 3 components replicate nicely, and even the relative ranking of the PCs is the same. Now, this is very interesting because now, rather than using 2.5 hours battery, I'm using a 15-minute battery that is kind of sampling the same domain, but you would think at a much coarser level, because I'm using only 50 minutes of testing, yet the patients, these are 140 patients in Padua, they kind of fall off exactly in the same. So as far as I'm concerned, now we're going to extend this project at the regional level. In Veneto, it's a highly structural health system. So we can have this project going in 26 stroke units in the region, and we're going to get data probably on 2,000 subjects. And hopefully this will replicate, because I think this is really the way you should think about stroke. If you want to do a stroke study for genotyping, phenotyping, epidemiology, treatment, thrombolytics, thrombectomy, I think you should look at these kinds of components, not just the NIH stroke scale, but really something a little better.
But you have lost explanation of 25% of your variance, more or less, right? So what's the kind of sensitivity that you would consider sufficient?
Well, whenever you do these PCs, of course, as I, yeah. So
Half full, half empty.
yeah, exactly. So it's 50%, in physics. I mean, in genetics, if you can explain, 5-7% of variance, you're happy. Here we got 50% of variance. I think in physics, you would say 50% of variance with 3 factors. I think is pretty good. But I agree. I mean, it's not perfect. I mean, 50%.
This has to give you traction in terms of the treatment trajectory of those patients, right? And then the question is 50% sufficient to give direction to the intervention you want to plan?
Yeah.
And I'm not talking physics anymore.
No, right. I think in this paper here, we track the behavioral recovery of these patients, and and you can track very well in the recovery in these patients using these metrics correlate with outcome measure, for instance, through the stroke impact scale or other things that are measuring outcome. So, I haven't done a recovery analysis using single scores, right? From, using a particular score. But again, I think it is a bit artificial because if you go back here and you look at, these motor measures, for instance, right? So here they're a measure of range of motion, strength, coordination, function, okay? And, you're looking at both shoulder, wrist, and hand, right? Okay, if you go in with the regular story, you might say, okay, I want a reaching task and I want a grasping task, okay? Well, the correlation between a reaching task and a grasping task is like 85%, okay? And why? Because basically whenever I'm moving my hand, it's not just I have a reaching system being activated and a grasping system being activated, which is the way we've been doing things. It is actually that I have an 85% correlation between shoulder synergies and wrist synergies. And with 3 components, I can account for hundreds of hand movements. So I think the perspective has to change. I think we have to recognize that the processes, the way we dissociate the processes based on neurophysiologies or a few single cases, is not the way the brain is organized. There is much more correlation than dissociations, I think. And I think what this data is emphasizing are the correlation, basically. Yeah.
The other PCs, so PC 4, 5, 6, whether they're replicated, they might account for a small amount of variance, but they're consistent.
Yes.
Just onto a different type of patient.
Yes, but I think when you have 140 patients, the first, as you see, as it is now, if you go by PCA rules, I should not consider this one, which is accounting for less than 10% of the variance. So this is why I need to scale up this study to 2,000 subjects, because now I can, even this 8% is going to get 15% or 16%. So I need more subjects, basically, to explore these lower components. Okay, so I hope at this point that you are somehow convinced that there are, at the population level, okay, then we can discuss, these traditional syndromes, but at the population level, these phenotypes are good phenotypes. Then the next question is, what are the lesions in the brain? What are the most common lesions accounting for stroke? And I think if you Google stroke and you look at the picture, it shows up a cortical stroke. We have this idea the stroke are a cortical problem, okay? This is ingrained in the psychology of neuropsychology. But in fact, when we look at the distribution of lesion in the WashU cohort, and this is a frequency map of all the lesions that have been segmented by 2 neurologists on a T1 image, blah, 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 you see that the frequency distribution is basically actually in the white matter and in the basal ganglia. And this is well known in the Lausanne study from the 1970s on CT scan. Only 15% of strokes were cortical. The great majority are either subcortical or cortical-subcortical. And this is the first map, T1 map, that shows that kind of in a modern form. And when we replicate the Padua project, we also see the same thing. We basically see that the hot spots are in the white matter. And when you look at the distribution of cortical lesion, you get only 14% in our cohort in Padua and 13% in the St. Louis cohort. And basically you get the same amount of subcortical lesions, 62% and 63%. So now these are 250 subjects. And so again, it is important to realize that when we study stroke, we cannot really think of cortical lesion or damage of nodes. We should really be thinking about damage of connections, okay, and modulatory system that getting into the cortex.
What do you think is here of the survival bias? I mean, because this kind of reminds a bit of this kind of old studies from the Second World War, where they mapped the hits on the plane, and they showed basically that the wings are most hit actually, and there was a kind of clear survival bias that planes were hit in different locations, just didn't arrive to the airport, so they couldn't be measured. Could it be that the stroke proportion actually just being biased, that basal ganglia strokes are least deadly, for example, and that patients are
No, there are biases here, but it's not a mortality bias. I mean, the percentage of mortality in this data set is, in general, the mortality in patient is rapidly decreasing. We're down to, in good centers, we're down to 8% or 7%. Okay, so it's a small fraction. Usually we don't study anybody that is in coma or very severe because they cannot do the tasks, but those people usually have huge hemispheric strokes. They're really not a single lesion, they're very large strokes. I mean, the biases here are the biases of the two biases. One, that middle cerebral artery distribution are much more common than other strokes. So you have mostly the center of the brain being affected, the MCA distribution, much less in the anterior cerebral artery, much less in the posterior cerebral artery. That's one bias. And the second bias that, for instance, in St. Louis now we have an 8% thrombolysis rate in which we give drugs to open up the blood vessel. In Padua now we have almost a 50% thrombolysis rate. So we tend to get only basically the most core MCA distribution. So we don't get a lot of, but I think again, when you look at the 1970 Lausanne database with CTs in which they didn't have thrombolysis, the percentage of cortical stroke only were still the same. So I don't think that these two biases that I just mentioned account for the fact that we don't have as many cortical strokes in this database. Okay, we have to move on otherwise. Okay, so based on what I told you then, it is not probably surprising that as we look at the number of correlated deficits in a single subject, if I say, okay, does this subject have one deficit, very modular, or does he have a lot of deficits? It turns out that there is a statistical relationship between specific regions in the white matter and the number of deficits that you have. So in regions of high traffic, in which there is a lot of white matter tracts, and these are these black regions here, and these are regions in which you have only white matter tract, you have only U fibers here, so it's color in white. This color scale gives you the number of correlated domain impaired. And so this area, the regions in which you have a lot of correlated deficit, fall in region the white matter. This is a very interesting prognostic implication, because now we know that if you have a lesion in the cortex, for instance, you're more likely to recover very well. If you have a lesion deep in the dorsal white matter, okay, in the corona radiata, where you have fibers going down the corticospinal tract, you have the superior longitudinal fasciculus and the arcuate fasciculus going anterior posterior, and you have callosal fiber going left and right. That, what we call convergence zones in the white matter, is a critical region for stroke to cause multiple deficits. Now, what about structural connectivity then? Okay, right now I talked about lesions. What about structural connectivity? So we've been moving into the structural connectomics approach, and right now we've been doing what is called indirect structural connectomics. Okay, in other words, we take a atlas brain of a this is the MGH atlas. Okay, these are like 1,000 DTI data sets, and basically we create tracks among the parcels in which we divide the cerebral cortex. These are the regional parcel derived from the functional connectivity. And then we can derive a structural connectome, which is basically the number of streamlines or probability between different parcels. And these are the networks. And so you can see this is a typical structural connectivity matrix, which looks very different from a functional connectivity matrix. So these are the subcortical nuclei. They're highly connected with all the cortical regions. And you see here, this is what is called a small world structure, where you have a lot of local connection and a few distant connections. And so we can get individual structural connectome by taking a lesion and embedding it into this atlas and extracting the normal streamline that have been affected. And then we can do group maps across different subjects. So we've been doing this kind of exercises, and you get interesting pattern of disconnection. So this is, for instance, one cortical region location, and you see you have a nice disconnection of the corpus callosum, and you also have intrahemispheric disconnection. Here is a lesion of the retrosplenial cortex, and this is the pattern of structural disconnection that you see. And you see, as you march through, you see kind of the structural disconnection patterns that arise out of focal lesions. Now, of course, think about all the lesion symptom mapping that we've been doing over the years. And I think you realize that, of course, there is much more complexity to that. And so to say that a particular voxel is specific for a particular function, it really doesn't make any sense anymore. And what I told you before about the convergence zone is illustrated here at the single case. This is a lesion in the dorsal corona radiata, and you see how much of brain is actually putatively disconnected by this lesion as compared to a focal cortical lesion that also show a fairly large pattern of disconnection, but not as comparable to a white matter disconnection pattern, okay? Now, these are putative disconnections because these are estimated based on normal atlases, okay? Now, we're just in the process now to measuring the disconnection in each patient, but of course that is much more complicated because of all the problem with DTI that you're well aware of.
There's two things here. How do you exactly assess the location and extent of the lesion? Do you reconstruct this automatically, or do you do it by hand? I think the second thing is indeed that the DTI assessment is an incomplete graph. Right? So how do you fill in the missing pieces of the DTI graph?
What do you mean incomplete?
Well, the DTI will not get you all the long-range connectivity. There's a resolution limit to what you can resolve by the DTI. And this might again lead to misinterpretation. So the question is, how do you get into the right shape exactly of this lesion? And how do you get now?
So the lesions are segmented. All right, so I mean, this is what we do. The lesions are segmented at the chronic stage. Okay, so when you have basically a better estimation of the lesion itself, they're segmented on a combination of T1 and T2. Okay, so you get also, and we're also segmenting the edema, if there is edema. Okay, so we're not just segmenting the structural damage, but also the edema. This is a probabilistic atlas of 1,000 subjects. So you're just, those fibers are basically the ones that are statistically more likely to be present in 1,000 healthy subjects. Okay, so with the limit of this data set, which is obtained with this super duper, 3 Tesla scanner with the 7 Tesla gradient, which is what they collected at MGH. I mean, this is probably the best DTI data set available in the world right now, with some exception, of course. So, I mean, at the population level, it's a good data set. But, I agree. I mean, this is problematic because, again, we don't know whether these are all the fibers, and we don't even know whether actually these are the real connections that are affected by that particular lesion. We should measure it in directly and then compare direct versus indirect estimation of structural disconnection and see whether the two are actually matching or not. Yeah. Right. But even if you directly measure it, it doesn't mean that you're getting, it is not clear that directly measuring the structural disconnection, it's better than estimating it in the atlas, to be honest. That's the real question. Right. Okay, so I told you about the behavioral variability, which is low dimensional. I also told you about all these lesions. They tend to concentrate in the white matter. Now you might ask, okay, how many patterns of disconnection do we have in the white matter? Does everybody with a lesion yields a very peculiar pattern of disconnection. And again, so you can do a similar analysis of structural disconnection patterns across many subjects, and you can do a PCA across many subjects. And this has been done by work by Michel Thibault de Schotten and Nashkef Barakesh in London. He has a data set of 1,300 subjects now. And he basically was able to show that with 7 patterns of disconnection, which are these patterns here, these are the first 3 patterns. With these patterns of structural disconnection, you can account for with 7 patterns, you account for about 75% of the variability of structural disconnection, okay? So again, this is saying that although you can have many lesions, 1,300 lesions, and they don't give rise to 1,300 pattern of structural disconnection. They give rise to a low-dimensional pattern of structural disconnection, just because the way the white matter is set up. This is very interesting. This is very counterintuitive, okay? This is not the way we're thinking about this. All right, so first summary slide is that at the population level, neuropsychology is simpler than we thought, okay? White matter damage is underestimated in stroke. So if you are a neuropsychologist, and I think what we've been studying in neuropsychology is what we call the tip of the iceberg. All right. Yes, you can find patients that have specific deficits, but those patients usually also have other deficits, and the double dissociation typically are tiny little things on top of big means that are the same. So you're looking in a double dissociation to the tiny difference, and we're looking at the common variance, okay? So I'm looking at the bulk of the iceberg. So a few behavioral clusters represent the bulk of the behavioral variability. They should be used as phenotype for structure, function, recovery, genetic, and intervention studies. White matter damage is common in stroke, low dimensional in topology, and predicts poor recovery. Hence, the secondary physiological disruption of cortical-subcortical network activity must be a major pathogenetic mechanisms in stroke. In other words, what's going on in the cortex for these lesions that are disconnecting the white matter is probably also should be accounting for behavioral variability, okay? Now, I think I've been very slow here.
No, it's fine.
No, but I mean, how much time do we have? It's already 11, so we don't have that much time.
No, it started a bit late, so it will be fine. Just
Okay, all right.
One question. I mean,
Yeah.
it's quite puzzling and very interesting that how it's so low dimension, white matter disconnection. Do you think edema is playing quite a role here? I mean, if you're in the anterior part of the corona radiata of the posterior part, you should get quite different effects. You could have edema.
No, stroke usually does not give a lot of edema. I mean, there are 20% hemorrhages in here, and hemorrhages do cause edema. But typically, you don't get, it's not like a tumor. A tumor could give you massive edema that might distort structures. And so no, I think this is really, and all the Barakesh lesions are chronic lesions, okay? So these are like chronic lesions in which the edema is subsided by then. So it is really puzzling. I mean, it is very puzzling. Okay, so now I'm going to tell you then what are the patterns of functional disconnection by looking at fMRI. And I'm going to ask the question, what is accounting for behavior? Are lesions accounting for behavior, or is fMRI patterns accounting for behavior? And again, what you need to keep in mind is how many fMRI patterns you might expect based on these different lesions. Okay, are we talking about high dimensional pattern or low dimensional pattern? All right, so the questions are, what are the physiological abnormalities produced by focal damage in stroke? What explains behavior, lesion, or functional damage? And this is all work by Josh Siegel, who's an MD PhD student. Okay, so how many of you know about resting state fMRI? Majority. Who doesn't know about resting state fMRI? Nobody. Okay, well, so then we can skip this slide. So, it's a temporal correlation of the Bolt signal over time. And you can then create correlation matrices based on temporal correlation. You can do multiple regions. We use particular atlases. We use the Gordon-Lohmann atlas from 2016. Now we're moving over to the Glasser atlas. So this is a matrix of the resting state networks. Those are the networks. You can project the networks on the cortical surface. And the first question that you might ask, okay, well, if I look at the matrix of correlation in my stroke patients, I mean, do I see alteration in my network structure? And the answer is no. Those matrices are about 95% similar, okay? So that's interesting. But it's also not surprising because the lesions are in different places, okay? So in different places, I'm taking out some connection and some patterns. And so overall, I think the mean remains the same, although, you know, there is no difference. Okay, so now we can ask the question, and we have individual lesion patterns, we have individual connectomes, and now we use something called ridge regression, which is basically kind of a multivariate analysis to relate this to behavior. So for every subject, we create a behavioral model, okay, based either on lesion or functional connectivity. Okay, and then we can look at how much of the variability is accounted by the lesion and how much of the variability is accounted by the fMRI pattern. And we do this for all these different domains. Okay, so for each one of these deficits, ignoring that they're correlated to some extent, now we're looking at single domain prediction. All right, and so we can play these kinds of games. So this is a model in which now this is the actual score, and this is the model score. Okay, so I see here that I'm predicting about 30% of the variance, and these are the individual scores across the different subjects. And so ridge regression allows me to back project my weights. So these are the weights that are predicting. This is a multi-domain score. These are the common performance deficits across all domains. Okay. In other words, if I have deficits across either any of these domains, I have, you know, this is my score here. And these are the weights. And you see here all these lines going across the hemispheres. And this is one of the main messages, is that whenever you have disruption of interhemispheric correlation, you have damage in your behavior. You have behavioral abnormalities. And almost it doesn't matter what is the behavior. I'm going to kind of say that a little more precisely. But keep in mind that the interhemispheric correlation at the acute stage is a major mechanism of disruption at the functional level. And then we can do all sorts of analysis. We can organize our connection by type of connection, for instance, intrahemispheric or interhemispheric. We can organize them by network. We can project those weight on the surface. I mean, I don't have the time to go through all these models, but you know, the pipeline kind of gives you all these possibilities. All right, so now we go down to the question of behavioral prediction, okay? And I'm going to start with, yes?
8 behavioral domains.
Yeah, I did.
Maybe I missed the definition of it.
So this is a word, I think is an average weighted z-score sum of all the deficits across here.
How do you define the behavioral domain?
Oh, the domains are the one that I defined earlier, these ones. The domains are the way I classify my tasks to begin with.
Ah, right, okay.
Language, motor, visual field, spatial memory, visual memory. Yeah.
Numbers of the domain of related to the measures you have done.
Yeah. These are, yeah, you know, if.
You had 7 there.
Yes.
So now you are using 5.
5. In this analysis, we also have, okay, we didn't do visual because visual, we didn't have enough subjects to do this analysis with visual. We don't have enough subjects. And attention, we did all of them, but attention in this case, I'm not looking at sustained attention. I'm just looking at egocentric neglect. Okay, so but these are like, I'm just for presentation purposes. In the paper, we got all of them. Okay, so, all right, let's look at language here. Okay, so let's take one, focus on one, and then I'll give you the gestalt so you get the big picture, okay? So language, this is, first of all, this is the variance accounted for by lesion is in white, okay? And functional connectivity is in black, all right? First look at the y-axis. I'm accounting for 50 to 60% of the variance of language score. So the model is actually doing pretty well, both the lesion model and the functional connectivity model. okay? And this is now the scatter plots. So these are real deficits, and these are predicted deficits based on this information. And you see, these are very nice scatter plot predicting high percentage of variance. Now, these are R-squared. These are not correlation, okay? These are R-squared. So the correlation is much higher here. Okay, and you can say here for language, both the lesion and the functional connectivity are predicting variability. Okay? That's one conclusion that you can draw.
But we need to know then how language is exactly assessed, right?
Language is the right. If I do a principal component across all the language scores, all my language domains, as I didn't say, but I'm going to say now, I don't get the regular syndrome that you might expect. I get a single language factor score that accounts for 75% of the variance in language deficits.
Okay.
And that's what I'm plotting here. Yeah, so it's an overall language deficit. Okay? So then let's look at the cognitive deficits. Okay? So this is egocentric neglect, this is visual memory, and this is verbal memory. And you see here that for lesion, as you look into memory, the lesion information accounts for very little amount of variance. Now, that is significant, but is not very high. In other words, to say that if I have a lesion in the brain, I cannot tell you reliably whether you have a memory deficits or not. Okay? Functional connectivity, on the other hand, is doing very well. It's still predicting between 40 to 50% of the variance. And when I look at the scatter plot, that is evident. You see here that the regression for memory is very bad, okay? But the regression for functional connectivity is quite good. 42% for visual memory, for verbal memory. So you might say that cognitive deficits are better explained by functional connectivity as compared to lesion. In contrast, if I go to primary sensory and motor deficit, visual and motor, I get the reverse. I get a much better prediction for lesion than I get for functional connectivity. Okay? Particularly for visual, my functional connectivity abnormalities do not predict much. So you see here there is a dissociation between primary sensory motor domains and cognitive domain in what is predicted by lesion versus functional connectivity. What is particularly interesting is that if you think about what are the weights that are predicting these patterns, okay, in terms of the functional connectivity, you might expect that every domain of this function might have its own circuit, might have its own organization, okay? Instead, what we find the same phenotype, and I'm losing you guys because I see people yawning and stuff. I'm gonna stop here. The same functional connectivity patterns are the same across multiple domains, but what is changing is where these connections are. So if you look at this picture here, this is the wedge, the weights of the functional connectivity for motor, left motor, right motor, visual, left visual, right visual. You see that the edge that are predictive, these are the predictive edges. There are always this interhemispheric disruption and this abnormal correlation within the intrahemispherically, okay? So these are the two major patterns, but what is changing is where these patterns are. For visual deficits, they're mostly within the visual network. For motor deficits, they're mostly within the motor network, but the patterns, the kind of connections that are affected are the same. So this is the good news, okay? There are this kind of a half empty, half full story. So on the one hand, we have a very low dimensional functional connectivity phenotypes, disruption of interhemispheric correlation, and abnormal correlation within the hemisphere. The phenotype is always the same. The bad news is that you have multiple connections that are affected. It's not just one region or one edge. There is a lot of connections that are being affected by these deficits. And when you look at attention, visual memory, and verbal memory and language, again, you see the same story. You see disruption of interhemispheric connectivity and abnormal integration within the damaged hemisphere, okay? And again, what is now worse is the number of nodes that are showing this pattern. So if I look at it at the network level, attention, neglect, about 60% of the brain is out of sync, and I have multiple networks being affected. If I have a motor deficit, it's a much more restricted part of the brain that is affected, and is mostly localized within the motor system. Okay?
There's something I don't understand here. So red means something else than green in terms of edges.
Yeah, red means increased correlation between increased correlation.
Green is less.
Green means decreased correlation. Now, these are the way they're coded that way because they're predicting good behavior. So if you have strong interhemispheric correlation, you have good behavior, okay? And if you have weak intrahemispheric correlation, then you have poor behavior.
Okay. And you said that for the motor and visual domains, the interhemispheric connectivity shows the same pattern. Right?
Well, no, the patterns. I mean, so here's.
That's just what you said.
No. I mean, the pattern. They're always interhemispheric. The location, though, is changing. Okay. The location you see here. This is a glass view of the brain. The visual cortex is back here, so the interhemispheric.
You said the same.
No, I didn't say the same. Similar in terms of the kind of connection they're affected.
Yeah, but then we have to quantify that. So if there's some graph theoretical analysis of that tells us quantitatively that this is the same.
Okay, no. So, all right. So, yes, good question. I need more explanation, which requires more time. So, I mean, I have a matrix here of 354 square, right? So there is a lot of connections. 354 parcel, square is a matrix. Okay. So in order to, understand what's going on here, we simplify and we divided the connection in interhemispheric connection. There are increases, okay? Increase interhemispheric connection, decrease interhemispheric connection, increase intrahemispheric connection, decrease intrahemispheric connection, and increase intrahemispheric connections. Okay? So this is kind of the
Sum, like a sum.
Yeah, it's kind of a sum metric. And so these bar graphs here are showing you for each domain which of the connection, according to this particular metric, are significantly related to behavior. Now, the green bar are the interhemispheric decrements. And you see for each domain, the green bars are nearly always the highest one. And this is the sum across, this is the 8 model mean that I showed you before, okay? Now, the next one are the intrahemispheric increases, okay? And the next one is the intrahemispheric decreases, okay? So now if we go into the weeds here, the intrahemispheric increases are particularly important for language, okay? Language is the only domain in which if you lose intrahemispheric connectivity, you have more deficits, okay? In all the other domains, usually is an increase of connectivity, and it's interesting, you've done EEG, and I think, it's very interesting. What happens is that networks that are segregated, okay, take the dorsal attention and the default network, are the first component in a principal component analysis of resting state network. Okay, the dorsal attention and default mode network, they're typically anti-correlated if you do global signal normalization. In a stroke patient, they start going together. So they're abnormally correlated together. And I think in the EEG world, this corresponds, and this is a speculation, corresponds to delta rhythms that you see in the damaged hemisphere, where the whole damaged hemisphere is showing delta rhythms. And I think this is what probably we're seeing here with fMRI. Okay, so now, I hope the main message here that is important to take home is that the phenotype of connectivity is the same, okay, it's low dimensional, but unfortunately where this phenotype is showing up is actually highly dimensional because we have many edges that can be affected, particularly for cognitive domain. Now, you might ask, okay, why do you see this different? Why sensory motor deficits are best accounted by lesion, and why cognitive deficits are best accounted by connectivity, okay? And I don't know why, but here's an explanation. This is what is called a spring-embedded graph of a normal brain. So basically, this is the visual network, okay? This is the motor network, and these are the cognitive network that you know very well, okay? Now you see that the cognitive network are more centrally located in the graph. The cognitive network are also much more connected by association pathways, while the sensory motor networks are much more connected to the periphery, either the retina or the corticospinal tract. So I think one of the reasons, a lesion in the motor system, particularly in the corticospinal tract, is going to give high prediction of motor deficits and much less the connectivity, is that if I knock out the corticospinal tract, there is no way for the cortex, no matter what it's doing, to connect with the spinal cord. So the motor deficits are best explained by where is the location of the lesion. The same in the visual system. If I knock out the geniculocalcarine tract, it doesn't matter what the visual system is doing, connectivity-wise, I cannot recover my vision. On the other hand, in this system in which there is a lot of redundancy, let's suppose that I knock out here a combination of default mode and frontoparietal, you see how many connections, indirect pathways that I can go through in order to recover those deficits, and therefore is the pattern of functional connectivity that matters. Okay, so this is an explanation for why we see this dissociation in behavioral prediction. Okay? Okay, so second interim summary, lesion location accounts for behavioral variance, especially for motor and visual deficits, but the mapping between lesion and behavior is not simple. I did not show you that, but it's basically, there is not a one-to-one mapping between deficits and location. Low-dimensional FC changes account for large amounts of variability, especially cognitive deficits. And the difference in prediction of lesion versus FC may reflect the normal organization of brain networks and white matter connections. I think I should stop there. There is another chapter, but on the relationship between structural connectivity and functional connectivity abnormalities. But I think we need a coffee.
Well, maybe we can hear the short summary of that, to at least get the flavor of it.
Okay. All right, so that's the question. What is the structural basis of functional connectivity abnormalities in stroke? So basically the question here is, okay, we have done the prediction of behavior. Now we have all these functional connectivity patterns, and we want to ask, what is explaining the functional connectivity patterns? Are the functional connectivity patterns explained by the lesions, or the functional connectivity pattern are explained by the structural connectivity, okay, abnormalities? And what is the dimensionality? I cannot tell you that already, the dimensionality of the structural connectivity abnormalities, okay? I mean, do we have many structural connectivity abnormalities that are predicting many functional connectivity, or a few, okay? The bottom line of all this little, I'm gonna show you, is that this low dimensionality that I'm describing here, when you look at the covariance between structural connectivity abnormalities, those 7 patterns that are accounting for 70% of the variance in Michel-Thiebaud data on the 1,300 subjects, in my functional connectivity abnormality, when you look at the covariance between those 2 patterns, it collapses even less. In other words, for those of you that knows 2 latent variable, okay, which is the structural functional covariance, account for 60% of structural functional connectivity variability. In other words, if I have a lesion in the brain, that lesion causes a low-dimensional pattern of structural connectivity abnormalities, probably 7 patterns, 75% of variance, which in turn causes a functional connectivity abnormalities, of which I show you the 2 main patterns. But if we look at the covariance between those 2 patterns are accounting for 60% of the covariance. Okay, now I'm going to show you, the bottom line is I'm going to show you this pattern, which is this pattern here. Okay, so on the left here, on the right here, I show you the most common functional connectivity abnormality patterns: disruption of interhemispheric correlation and abnormal correlation within the hemispheres, okay? And each one of these nodes is colored by the network. I'm sorry that the picture is a bit fuzzy. This is the structural connectivity pattern that is strongly predicting this and this. So the structural connectivity pattern that is strongly predicting those two functional connectivity patterns is the loss of callosal connections. And when I look at where in the white matter this is happening, this is where it's happening in the white matter. It's perfectly matching the hotspot of the lesion analysis. The hotspot of the lesion analysis corresponds to the regions in which you're losing callosal connectivity, and therefore that causes the functional connectivity abnormality that I've shown you, which are in turn explaining behavior. So there is a cycle here. It's a chain of events that is closing perfectly. We got a lesion. The lesion is disconnecting the corpus callosum. Depending on where that happens, that causes different patterns of network abnormalities, but the pattern is always the same. And depending on where that happens, explains the behavioral deficits, either sensory motor deficits or cognitive deficits. Now for treatment, of course, this is very good news. Why? Because, again, it's very good news and it's very bad news. It's very good news because if I can fix this pattern, okay, if I can fix this pattern, I can fix 60% of the behavior. All right? It's very bad news is that because all our attempts to do single-site TMS, in my view, are hopeless, or TDCS. Why? Because we're just stimulating a small part of brain. And the problem is that we have to find a smart way to reactivate these patterns with multi-site stimulation, if we wish. Because now you have to hit specifically in each particular subject those specifically set of connections that are specifically damaged by the. Now we know what they are, but the edges are going to be slightly different in each subject. Okay? So that's the bottom line of the talk. Yes? I'm sure.
Structural damage doesn't explain the variance for specific kind of behavioral deficits, and it does for others, whereas you find the opposite in functional connectivity.
Wait, structural connectivity?
Yes, structural connectivity.
It does explain variance. I haven't shown you that.
Okay, because this is kind of heading towards that point.
There is one little bit of, yes, but
okay.
That's another 30 minutes. Yeah.
Okay.
So we do find very strong correlation with structural connectivity and behavior. Yes. Everybody's scratching their heads.
Shall we continue with our question session and now? This for your conclusion?
Yes, I mean, yes, I need to show you my collaborators. I think that's very important because this is not a one-man show here. Okay. So it's not that many people, but so these are all the postdoc. Antonello did some of the first functional connectivity analysis. Lenny did all the recovery work. Nick is our biostatistician. Joe is the new postdoc that is doing all the structural functional connectivity abnormalities. Chiara is doing some of the modeling along with Mohit, which I did not show you. And Josh has done all the lesion fMRI predictions. And Gordon Schulman is my good friend and collaborator. And for the last few years, I've been collaborating along with Gustavo Deco on the modeling of all of this.
Okay, very good.
Yeah.